Hello everyone, welcome to exam topic covered in 3 minutes, and in this video we are covering the very important topic, that is variants of PCR. So the first one is reverse transcriptase PCR, that is what we call as RT-PCR. And what's the function of this? This is to amplify any types of RNA. And what are the steps that we use? The first one is it requires a reverse transcriptase reaction, so that what we produce is called as a complementary DNA, which is otherwise called as cDNA. Now, using this double-stranded cDNA, we are using a usual PCR technique, or we are doing a very usual PCR technique, and one enzyme like Taq polymerase in the case of conventional PCR, what we use here is TET polymerase. The advantage of this enzyme is it has two activities, DNA polymerase plus reverse transcriptase. Now, what is the use of this reverse transcriptase PCR? It is something to study the RNA, so it is a gene expression study. The next variant is real-time PCR, otherwise called as qPCR. Remember, this is not RT-PCR. What is the function of this real-time PCR or qPCR? It is to quantitate the DNA, and this is during the exponential phase. So what is the technique that we use to quantitate? Number one is we use certain intercalating dyes like ethidium bromide. Then what we use is SYBR green. So this is the picture of SYBR green, and we use certain probes which are called as Taqman probe or molecular beacon, etc. Right. Now, what are the applications of real-time PCR? First and foremost is to quantitate DNA. So if we add a reverse transcriptase reaction with this real-time PCR, then it is called RT-PCR. Then we can quantitate the RNA as well. So real-time PCR is to quantitate DNA or RNA. That is very important. Then another thing is that this is used to detect the length polymorphism or mutations that affects the length. So these are the uses of real-time PCR. Next, another one is nested PCR. In nested PCR, basically we use two sets of primers, that is outer primer and inner primer. And when we do nested PCR, if the DNA sample that we have is very low, so the technique here is we will do a first set of PCR using the outer primers, and using the amplicon of that, we are using the next set of PCR, and that is using the inner primers. So what is the advantage of nested PCR? It is having high sensitivity and specificity. The next important variant is multiplex PCR. What is multiplex PCR? Here it is like a multiplex theater where multiple cinemas are played at the same time. So just like that, we are adding multiple random primers. So we have a set of DNA inside the mixture. In the same reaction mixture, we are adding multiple set of primers for set A, set B, set C, like that. So that we will get multiple amplicons, and each will be having a unique fluorescence. What is this approach called as? This is called a syndromic approach, a very important previous year question. So this is used in the diagnosis of certain disorders like meningitis and pneumonia, where we want to get a rapid result, like which strain or which bacteria is infecting the child or the patient. So if we want to get a rapid result within a matter of 1 hour, what we use is multiplex PCR, where we add multiple primers of different bacteria, and if the bacteria is there, we'll get the amplicon very soon, and we can find out what is this bacteria infecting the patient. So this is called a syndromic approach, which we use in multiplex PCR. So these are the important variants of PCR. See you in the next video. Bye bye, and follow for more.